Since this is the computer scientist group, I'm not sure how familiar you are with deep learning and like CNNs, basic CNNs. Would you all say that you're completely familiar with that, how that works? Did you do some deep learning stuff so far? Yes, yes, maybe, no. Well, we go over some of the basics, okay? Just to make sure we're on the same page. So this is kind of the traditional CNN architecture that was first popularized. So essentially we have some input on a grid, and what we're using are these convolutions. I'm going to explain those in more detail later. To create feature maps with more and more abstraction, we pool in between, create more and more feature maps, and in the end, at least in these traditional architectures, we would have a fully connected layer and go into some output classification or whatever we are doing here. So this is how convolution kernels work. This looks quite complicated, but I'm going to walk you through this. So we're going to have an input volume here. This input volume has 3 channels, so we have 3 different versions here. Okay, you could imagine if we have an image input, this would be the RGB channels of the image. Okay, and then we have pixel values in here, if we stay with this metaphor of the image. And we define now convolution kernels with a certain size. The size is the first property. Hi, do you want to join? No? Okay. So this is the first property of a convolution kernel, is the size. So in this case here we have 3 by 3 convolution kernels. And an important property is that these are always going through the full depth of the input volume. So if I say 3 by 3, I implicitly say 3 by 3 by 3 because this is the input channel size. Okay? We're going to have weights. These are the numbers here in red. And these weights are going to be the same no matter where I place my convolution kernel. This is the other important property. So I run this convolution kernel over the full input, all possible positions. Hello? So we run this convolution kernel over all possible positions and multiply with the weights, add a bias term if you have that, and this is going to be the output. So this is the output volume for the first filter. In total we have 2 filters here, so each filter leads to 1 output map. So in total here, using 2 filters, we have output channels 2. Okay, so we have 2 feature maps generated by this. All right. Now the other things that we need to define when we want to use convolution kernels are padding and stride. Now the padding is the amount of, or rather the layers of zeros that we want to pad around the input image. We use that to make sure that we can use larger kernels like this on positions like here. If we wouldn't do any padding, we would only have 2 possible positions on the horizontal axis to place this kernel. Okay? So this is kind of for the control of the size of the output maps. And the other parameter which is in relation to that is the stride, which means how many positions do we skip when we walk across the input. And this could be just 1. Stride of 1 means we do not skip anything, we use all possible positions. Okay? Anything else about that? This is kind of the basics of this. Now, an important property, and we will come back to that later when we go to graph convolutions, is these filters are always rectangular. And this makes sense because we have grid-based inputs. And we are using the specific topology of this grid base because in an image, for example, neighboring pixels are not really independent, they are kind of correlated, and we're using this kind of information with these convolution kernels. And later on when we get to graphs, we'll see an extension of that. Now in terms of the sizes, there's a nice formula here. So if you have some input size given and you want to have a certain output size, you can basically compute it here by setting certain kernel sizes, padding and stride. And what we oftentimes do if you do not want to pool or kind of shrink the input dimensions, we want to conserve the input size. For example, I'm not sure if you were in protein prediction 1 in the summer semester, I talked about contact prediction, and in this case we have an input with size L times L and some feature channels, and we have an output which is also L times L, so we do not want to modify the width and height at all. We want to conserve this throughout the whole architecture. And then we can use combinations like this. So for example, when you have a kernel with 5 by 5, depends on what kind of receptive field you want, you can use a padding of 2 and a stride of 1, and you will conserve the size. It's going to be the same in the output for that layer. Okay? Now in terms of architecture, so in deep learning, as the name implies, deep, we want to have multiple layers. So we have many of these hidden layers stacked on top of each other. So this is kind of, if you want to read this, it goes from the top downward. So this is basically how you would do a convolution layer in a modern CNN. So you have this convolution operation, as I explained it previously. Then you have batch normalization. This is just for stability, numerical stability, for the gradient flow. And you have some kind of activation function like you have in any kind of neural network architecture. This could be a ReLU, it could be 10H, it could be sigmoid, whatever you want to use. Usually some kind of ReLU. In this example I've chosen preLU, but it could be anything else. So what you do then is you stack up multiples of these layers. So for example, here in this architecture, which is one I used for contact prediction, you have some input layers basically, which are feature compression layers, where we just bring down the number of feature channels to something more manageable, just for memory. And then we have the actual backbone where we stack multiple convolution layers, in this case 5 by 5 with 64 feature channels, size conserving in this case, so it's going to be the same output size all the time. And then we have some kind of output layer, in this case here this is just a binary classification that we do for basically each pixel value. Now, there's another set of architectures which is very important, and these are autoencoders. So these have been around for quite a while, and they are still fairly recent, so this is still used. At least some variants of those are still used. So essentially what you have here is an architecture where you have an encoder part. So you start with your input, you feed it through the encoder part, through these input layers, until some latent space representation here. Then you basically blow it up again into an output layer, and this is kind of a mirror version of the encoder in this decoder part. And the goal usually of these autoencoders is to reconstruct the original input, which might seem weird at first. Why would I want to do this? I want to have the same output as I used as an input. But the important property here is we want to have an encoded version, a latent space representation that could be more compact. Now, actually this compactness is pretty important. What would happen if this latent space here would be as large as the input and output sizes? What could happen? And what would most likely happen? If we're trying to reconstruct the original input. Yeah?
The reconstruction error would probably be very low.
That's one way to phrase it, yeah. So what kind of function would the neural network actually approximate?
Sorry?
What kind of function would the neural network approximate?
A reconstruction.
Yeah, it would do that. I mean, the error would be very low in the end, that's true, because what it would most likely learn is just an identity mapping. Okay, if we do not constrain this latent space, it could just push through the whole input. Okay, so that's not quite the point. So it needs to be more compact. So it's actually encoding, it's kind of compressing it down. And this is actually one of the application scenarios, of the early application scenarios. It was used for dimensionality reduction, just like PCA, for example. And nowadays it's often used to get more compact representations of whatever it is that you want to do, like proteins, for example. Even though there are more sophisticated techniques to do that, and we'll briefly talk about that as well. Now there are some variants of these autoencoders, and one important one is the variational autoencoder, which has a very different mathematical foundation actually, but the overall architecture still looks the same. Now what people thought about was, well, what happens when we, let's take an example. Let's say we're training on cat images, okay? So we're training this autoencoder, and it's supposed to reconstruct the original input. We have some compact latent space representation, and we have some kind of, well, representation of cat images in there. And the network is able to reconstruct based on that. Now you could say, okay, what would happen if we do not use an input to encode something, but rather try to sample something from this latent space, and then try to create a cat image out of that? People tried to do that, and actually with this default autoencoder it didn't work at all. And the problem is it didn't work because we have no idea about the structure of this latent space. So we have no idea what kind of manifold is that. We can sample something, but it's mostly going to be garbage, and the network is going to produce garbage. It's going to just be noise in the output. Then this idea of the variational autoencoder came up, and essentially, as I said, it's the same thing. The only thing is we make constraints on this latent space. So the network actually models a distribution here. And if this is a distribution, we can actually sample from that in a meaningful way. And then we can start to generate images, or whatever we do when we train this. Depends on the domain. So this is kind of an early version of a generative architecture. This was before systems like generative adversarial networks came up. It worked quite well, but it was superseded by more elaborate architectures. Now, we have variants like the denoising autoencoder. This is basically a standard autoencoder, with the exception that we add noise during the encoder part. Now, we do this for several reasons. One reason is, oftentimes, especially in biology, we have noise in our data anyway, okay? And we want to make the system robust towards that noise in the data. So we don't want the network to basically overfit on the noisiness that we have on the inputs, okay? And if we add noise anyway, the network is going to learn that it needs to filter out the noise when it's doing the reconstruction in the decoder part. And here we don't add noise, obviously. We want to reconstruct the original input, but we add the noise in the encoder part. And this is quite a powerful technique. Now, if we use an autoencoder like this, it's going to be basically unsupervised training. We're creating embeddings in the middle, but there's no way to actually enforce a certain structure of the embeddings, except when using variational autoencoders, but that's a different goal. But what we can do is use something which is called ladder networks, which is basically a combination of a denoising autoencoder, where we also branch out from here. And we add an additional loss term. So let's say in bioinformatics we could say we want to do secondary structure prediction on this latent space. And this is kind of an additional loss that we enforce here, and we hope that by doing that we introduce some biological meaning into this latent space representation. And the hope is, of course, that we get embeddings which are then better suited for other prediction tasks. And sometimes this works, sometimes it doesn't, but it's a cool technique. Now, what we are doing here in the lab recently is using ELMO embeddings. This was covered in the lecture in the summer semester, so I'm just going to over this briefly. So essentially what we are using here is a technique which is borrowed from NLP, from natural language processing. Essentially we are stacking LSTMs in a forward and a backward direction, and those are supposed, at least in the language realm, supposed to predict the next word in a sentence, or in the backward direction, the previous word in a sentence given all the tokens before that. This is done with unsupervised training. It works fairly well, and in the end you get some embeddings which you can use for other prediction tasks. And in our lab, Michael Heinzinger and Ahmed El-Nagar have been doing that in the protein realm, which works quite in the same way. So what happens here is we are predicting the next residue in a protein, or the previous residue given all the others before that. And in the end, after training this model and bringing down the perplexity of it, we extract these hidden space representations for a protein, and we're using these embeddings as inputs to other prediction methods, and this works really, really well. And it's one way to maybe at some point even get around multiple sequence alignments and evolutionary information. At least this is our aim, which would be great. Now, in the realm of traditional CNNs, what most people converge to nowadays is residual networks. This is kind of the gold standard right now. So this is very similar to the standard convolutional architecture I showed you before. The only difference is that we have kind of another level of abstraction in here. So we have these residual blocks here, which are the basic building blocks of the CNN now. We have an arbitrary number of this. This is kind of a hyperparameter here, how many you actually use. And each residual block has this form here. So in each block you can have multiple convolutional layers, again with batch norm and nonlinear activation function, of course. But the trick here is that we are feeding this original input of the block through to the output, and we're summing those up. And the idea behind that, the justification, is that we want to conserve the less abstracted information and push that through the network all the time. So at the next block, what comes out of here is also transported to the output and added up. Okay? We're doing this with all the residual blocks, and this makes the network actually much more powerful, because it basically has access to all levels of abstraction at each point. This is the idea behind it. Okay? Yeah. So this is it for the standard CNNs, and now we're going to graph convolutions. Now the idea behind graph convolutions is really we just want to generalize this idea of CNNs. In CNNs, in traditional CNNs, we only have these rectangular filters, right? And for that we need grid-based data. If we don't have data on a grid, well, we are kind of lost. What do we do? The abstraction of that would actually be graphs. So this is some arbitrary graph. Graph in general is defined by a number of vertices or nodes and a number of edges or connections between those nodes, right? Now, as you can see here for a grid, this is also a graph. This is a graph with more constraints. It has a certain topology. So this is kind of a special case of any kind of arbitrary graph, okay? Actually, we could model it like this, but we could also include the diagonal connections, and we would need to do that if we want to kind of reproduce traditional CNNs with graph CNNs. But this is certainly possible, which also shows that this is kind of a more general way of looking at things. So we need some terms for graphs now. We are mostly working with undirected graphs, at least in this lecture here, but there are extensions for graph convolutions for other networks as well. So we are using undirected graph, which means that these connections are always symmetric. So if one node is connected to another, then that also means that the other node is connected to the original one. Now, an adjacency matrix that you can see here is basically a matrix modeling all these connections. So this is binary in this case. We're having a 1 for an entry when these two nodes are actually in contact with each other or have a connection, and 0 otherwise. And in this case here, for a default adjacency matrix, we ignore self-connections, so this is going to be 0 on the diagonal. Now we can have directed graphs. So the connections are not symmetric in this case, at least not in general. This also means that the adjacency matrix of this is not going to be symmetric anymore, obviously. And if you are looking at the degree of nodes, sorry I forgot that to explain that here. So the degree basically is just the number of connections that a node has. And you can easily get that by simply summing up the rows or the columns in the adjacency matrix, and then you get the degrees out of that. Hi. For a directed graph, as I said, this is not symmetric anymore. This is actually a special case here because we have a non-cyclic graph. If you would have cycles, you would also have ones here in the lower diagonal, in the lower triangle. But anyway, now degree becomes a murky term because now we have to differentiate between out degree and in degree, right? But it doesn't really matter for this lecture here, we're not going into that. Just to mention it, there could also be weighted graphs. In this case, it's a bidirectional weighted graph. If it's bidirectional, so no actual direction applied like here, the adjacency matrix is symmetric again. But now we are not having a binary case where we have zeros or ones, but we actually have the weights in there. Okay? So, one very important thing when we're talking about graphs is the graph Laplacian. Now, the Laplacian you've maybe heard in other contexts like engineering, maybe, or physics, like the sum of partial derivatives, and it's often used for smoothing certain properties in some field, for example. And the idea here is actually quite similar, even though the mathematical expression is quite different. But this is also used in spectral graph theory to basically smooth out properties along edges of a graph. And this is also the motivation for the idea for the graph CNNs, basically. So the graph Laplacian is simply defined as the degree matrix minus the adjacency matrix. And when I say degree matrix, this is simply a diagonal matrix which has the degrees of the nodes on the diagonal and zeros otherwise. Okay? I'm going to show you an example for that in a second. So yeah, as I just said, this is a mathematical formulation. So on the diagonal, if I is j, it's the degree of this node. And otherwise, off the diagonal, it's either minus 1 if these nodes are connected, or 0 otherwise. There's another formulation of this Laplacian which is called the normalized symmetric Laplacian, which is just computed in a slightly different way, either based on the original default Laplacian like this, or with the identity matrix and the original adjacency matrix. Basically it looks like that, so it's 1 on the diagonal if the degree is not 0, and otherwise it's this, this fraction here with the square root of the product of the degrees of the two nodes, and otherwise 0. So what does it look like? So here's an example of a graph Laplacian, the default graph Laplacian for the simple graph. So as you can see here, you kind of have the degree matrix still here on the diagonal. So these are the node degrees. And then you have a minus 1 for the connections and zeros otherwise. Okay, so now we're going into a little bit of mathematics here. We need to do that to kind of explain why we get to such a nice formulation in the very end. Essentially the goal is to have something which you can practically use in a deep learning framework where we can reduce everything to simple matrix multiplications, because this is also what we can parallelize. And now this is a bit of a lengthy process, bear with me, we're going through some concepts here, but it's going to be worth it in the end, and it's also important to explain when this actually works and when it doesn't. So, we're going into spectral graph convolutions. Spectral graph convolutions in the general case are defined like this. And this is motivated by the eigenvector decomposition of the normalized graph Laplacian. It looks like that. Maybe you remember this from linear algebra back then. Okay? So essentially U is a matrix of eigenvectors. This big lambda here is a diagonal matrix of the eigenvalues. Okay? And we're using this composition here, so we are introducing this function g, parametrized by theta. And this is essentially a function of the eigenvalues of this Laplacian. Okay? Now the problem here is, if we were to just use it like that, for big graphs, these eigenvalue decompositions are really computationally expensive, so we don't want to do that. So what we do is we approximate that, and there's a nice scheme from this paper here. If you want to look it up, all the details, then feel free to do that. I'm just going to go over this relatively quickly because we're going to approximate and simplify this even more in the next step. So essentially we can approximate this with Chebyshev polynomials. So this is roughly speaking the sum of our parameters times these polynomials here with this formulation. These polynomials, if you don't know what these are, they are defined recursively. And the important thing, the only thing really that we need with this is that t0 of some input is always going to be 1, and t1 of some input is going to be the original input again. This is all we actually need, so we don't even care what the rest of these polynomials are. Now if you put this back into our full convolution formula, we get to something like this. And now what we're going to do is we're going to set our big K to just 1. And we're doing this to just look at the first degree neighborhood of the nodes, because this is what this sum actually does. So it basically sums up contributions of the neighborhoods with certain degrees. So for 0 degree neighborhood, which doesn't really make sense, first degree neighborhood, second degree neighborhood, and we're restricting ourselves to just the first degree neighborhood. We're doing this for a specific reason, because then the whole thing simplifies a lot. So if we set the big K to 1, we're just having these two factors in here. So that's the first one here. The other one, this happens because, as I said, t1 of anything is going to be the original input, so this is going to be just this L with this tilde here. And we're also making the approximation that lambda max, our largest eigenvalue, is roughly 2. This is just a scaling operation. This doesn't matter in the end because the neural network weights will adapt to that. And if we do that, this actually cancels out. We just get these L minus I. So this is what you see here. Now we can even say, okay, I do not want 2 parameters in there, let's just make it 1. We just set the one to the inverse, the additive inverse of the other. Then we can push this out, and essentially what we get is this formulation here, which is also much cleaner now than what we had before. Now, this is slightly numerically unstable, so the original paper introduces another renormalization trick, which actually makes the formula much less bloated and very, very compact. So essentially what we have here is one big matrix term, somewhat based on the graph Laplacian. It's kind of a modified version, but it's based on the graph Laplacian. We have our weights and our original input. So, after all this math, what does this mean if we actually want to use some input signal? Let's say this is n times c, so we have n nodes in our graph with c features. So c input channels in deep learning terms, and we want f feature maps. Well, what does it look like? We have this big matrix term, and this is static for any graph. We can pre-compute this, and we should probably, okay? So we are pre-computing this matrix. So we have this fixed matrix, and all we're doing is we apply this matrix to our input vector x, multiply this with our weight matrix, and get our activations out, okay? So this is super simple, and this is parallelizable. You can implement this in a deep learning framework, no problem. So this theta here has size c times f, which is nice. So this is kind of like standard convolutions. You only care about the input channel sizes and the output channel sizes. And the same here for the output. So what we fed in was n times c, so n times the input channel size, and what we get out is n times f, so the amount of feature channels that we want. So this is pretty much exactly usable like a standard CNN, where you care about input, output, and the filter parameters, and we do not even care about filter parameters because in this case we're always looking at the first degree neighborhood. So, to phrase it in more deep learning terms, if you have some hidden state, hidden layer, HL, and you want to know what is HL plus 1, this is probably what you remember from standard neural networks. It's just, again, this kind of matrix here times the previous activations times our weight matrix, and of course we apply some nonlinearity, ReLU, or whatever. So in pseudocode, and there are actually libraries which are usable pretty much like that for graph convolutions now. So you have some GCN layer, just like you would do a conv2D, for example, in PyTorch. What you throw in is your original input, so your node features basically, and the adjacency matrix, and you get the next hidden state representation. Push that through a ReLU, and that's it. So this is pretty easy to use. So how can we actually apply this in computational biology? So the first thing I jumped at when we in the group looked at this paper was contact maps. Because essentially a contact map, a binary contact map, is an adjacency matrix. It is exactly that. So you have a 1 for the pairs of residues where you actually have a contact, where they are in close proximity, let's say 8 angstrom threshold, which is the default, and a 0 anywhere else. The only difference to a default adjacency matrix is that we kind of have these self-connections in the diagonal, but this is the only thing, really, that's different. Otherwise, we could say, okay, this is kind of a graph representation of a protein. Now we could say, okay, I could have some sequence-based features, maybe it's just the amino acid frequencies, or it could be ELMO embeddings or whatever, and then we use this in conjunction with a contact map as an adjacency matrix and push that through graph convolutional layers. And for example, predict some function. We could predict Go terms, for example. So I really got stuck on this idea. It didn't work out so well. Just to mention it, you could of course also use distance maps. This would kind of be a representation of a weighted graph, but it would work in the same way. Now, I was particularly intrigued by this idea because there's also a paper doing exactly that. Okay, so this is not peer-reviewed, it's not published, it's a bioRxiv preprint from 2019, but they're using exactly this concept. So they're using a contact map as an adjacency matrix for graph convolutions. In this case, they're also doing something which is very similar to our ELMO embedding. So it's basically stacked LSTMs to get protein representations. So this is their sequence feature that they are using. So using that together with the contact maps, using 3 of these graph convolution layers, go to some fully connected layers and then predict Go terms. This is what they do essentially. Now, I think this is not peer-reviewed yet for a reason, because this approach actually doesn't work at all. The reason is in the mathematical formulation. And this was not quite obvious to me because I was also not an expert in spectral graph convolutions. But the problem here is, and this was clearly stated in one review paper on graph convolutions, all of these spectral approaches, in all of those, the learned filters depend on the Laplacian eigenbasis. This was kind of the starting point, right? So this means this is specific for a graph structure. You cannot train on multiple proteins all the time because you need one big graph. You're not going to learn anything reasonable from that. So it doesn't work with contact maps. This doesn't mean that graph convolutions in general don't work here in this case. It's just that these spectral graph approaches don't work. So basically, you need to figure out something else. And there are different solutions for that. Now, this doesn't mean that we cannot use this concept at all for bioinformatics. We just have to be careful when we apply it. So this is an example from a paper. It's also just a preprint, not officially published yet. But this one does it right, because what they do is, well, they are doing many things. They're using these relation networks, by the way. I'm not going to go into detail here. But essentially what they use graph convolutions for are these protein-protein interaction networks. Now the difference here is we are not encoding proteins like graphs, but rather in this case we have one big static graph. And for this actually works because you can compute this big matrix term. It's going to be static for the whole graph. You can actually work on this because it's going to be the same graph all the time for the network. Okay, so for this one here, especially protein-protein interaction, I think the spectral graph convolutions are very, very useful, but make sure to not use them to represent proteins and use different proteins, hence different graphs in a spectral graph convolution network. Okay, so this was kind of my mistake. I was kind of investing into that direction, which also led to the lecture being a little bit shorter because the approach doesn't work. But at least now you know how those spectral graph convolutions are used and when you can use them.
So yes, let's dive into this. Well, just a brief introduction. Who am I? I like to think about myself as someone that kind of works through data. So this is me. I'm all over the place, I hope. And let's dive right into the problems that I usually see or that make me mad when I see presentations by people. So here we have a table of profits, and this is just numbers. I don't know, do you see something? If you don't, is this better? And if this is not better, is this better? So ultimately, can you turn the lights off maybe? Yeah, that one, perfect. So ultimately, of the three, which one would you pick by raise of hands? Who would pick the table? Who would pick the pie thingy? Who would pick the bar chart? Yeah. So there's definitely a preference in what is visually pleasing and the way you present data. And there's actually science behind it, so it's not just something that you have a feeling for, and design doesn't matter. Design matters a lot. And going in the direction of design matters a lot, what is the difference between the two graphs? Who has some kind of the willingness to tell me their feeling about these two graphs? We had this yesterday in a group meeting, so it made me mad. No?
Well, it's a colorful way to represent that. Like color also represent some threshold. Like if it is going above some threshold, then you have the different colors.
Okay, so his interpretation is that color represents threshold, but in this case there's no threshold. So you are automatically prone to associate that color means something. So color has absolutely no meaning, no reason to exist if there is no meaning behind it. Meaning that in this specific graph where there's no point of having the color, there's no point in putting the color. So please never put colored graphs if you don't need colors. Because color is information and information is interpreted. So yeah, that's a problem. Now, 3 seconds to look at this. Someone has something that he wants, what or she wants to tell me about this? What can you see?
It's a decreasing trend or something.
So you can see that the trend is decreasing in both graphs. It's 2 separate years. The trend seems to be the same, but there is a problem. There's a 10% loss with respect to the previous year because the graphs are not centered the same way. So one is at 100 and the other one is at 90. So if your goal is to show the trend, this is good because it's the same trend. Actually the data is the same, I just lowered everything by 10. But the starting value is 10% less than it was the year before. So you have to be careful about what you are showing people, and you can trick people into whatever you want if you are not paying attention or paying too much attention to some things. So if you read a paper and they present wonderful graphs, they might be good, but they might also just be there to trick you. And now this is another one that makes me particularly mad. Is there anything that you see here? What are your impressions? Just tell me. Yes?
Yeah, the color choice means that green is good and red is somehow bad.
That is one thing. And then the other thing?
The fitting, right? The graph fitting is much better than the green.
The graph fitting?
Yeah, means you have the random data, then you fit through a line, like a linear representation.
Mm-hmm.
So the red one is much fitting the data, but the green one is not fitting.
Yeah, I mean that is sure. I mean that's a thing, but that's not what I'm after here. Okay.
Ask Tobias.
Yes. So there are colorblind people on this planet, and to them these two lines are exactly the same color. The points are exactly the same color. So if there were no lines, they would just see a cloud of points of the same color, completely meaningless. So, and the second point that is very infuriating to me is more visible from this representation. So now I'm using colors which are friendly to colorblind people, and I'm giving you also error bars. So what is different from the error bars here with respect to what we saw earlier? There's no difference between the two lines in error bars. There's absolutely no difference. They overlap, so the data is not significantly different. There's no point in showing two different lines because the data falls into the same distributions. As simple as that. So this is a lie. And this is also kind of a lie. Now we get into another example of why sometimes visualizing data is useful, and I took this example from the web, otherwise I would have never put a red line. If you just look at one particular metric, which in this case is the Pearson's R, the 4 graphs that are presented to you have more or less exactly the same Pearson's are. If you start looking at the Spearman's rho, there's a discriminatory difference between the data sets. And if you would have never visualized the data, you would have also been never able to tell. So it is useful to present data and to visualize stuff. And you should try as much as you can to visualize data in many different ways before you make any assumptions on it. Because if you just visualize it in one way, if you just use one technique, if you just use one metric, you might miss out something in your data which you didn't account for. And this unfortunately happens very often in biology because we don't really know the ground truth of biology to a systematic extent. So we do know that the physical and biophysical properties of proteins and some of the and cells and pH and whatever, but there's at the systematic level just no clear way of understanding this. So if you look at an entire protein chain, this is a complex object. We cannot simulate it even. So let alone just represent it as a single value somewhere. So if you do this thing of representing data in single values, try to use different types of correlation matrices or just distance matrices. And now to some more, personal kind of things that I've done, and this goes in the direction of how can we show different things for the same data. I worked together with this professor in Singapore, who's now in Shenzhen, and he just was one of the co-founder of a new experimental approach which gives you the ratio of proteins at a specific temperature in a cell line. So if I put proteins, if I have cells, I heat them at a certain temperature, and then I look at how much of a specific protein is still in that cell at that temperature. And you can do this for different temperatures, and the higher the temperature is, the less proteins there are, because by nature it's a biological process by which the protein becomes unfunctional. So what this turns out to be is that you may have the same protein is represented by the same color in different experiments, and they follow sort of like a trend line, right? So at 38 degrees Celsius to 64, at 38, most of the protein in the cell is still present, and then at 64, the protein is almost entirely dead. And the same applies more or less to a different protein, but there clearly is a difference between the two proteins. You can see this from the graph already, from this trend line graph, but it's even more evident from the graph which uses just nodes and the graph basically, where the edges between the two are Euclidean distances, and the bigger the Euclidean distance, the further they are pulled apart, or the closer they are pulled together. So in the case of the two experiments on the same protein, they're obviously very close in space, they form a cluster, and they're pulled apart from the cluster of the other protein. So, this is still a very debatable project, because we still don't know exactly what or how many of these, how many of these different representations we want to include and which ones make sense. So what we did is basically to just add a bunch. So what you can see now is these are all fairly similar in their distributions over temperature. And then here you can see, they are all pretty close in space. So what if I start using different metrics on the distance in the graph? Well, you can see that there are some more outliers. So for example, this protein seems to be a little bit different from all the other ones, which is kind of true. So you can see that here, most of these, like this one has a sharp turn here. This one sort of like follows the blue one follows a sort of like constant trend, whereas this one has sort of like a shift in direction. It goes up again and then it goes down again. So, these are all things that you want to somehow represent immediately to people without going into the details of the math of derivatives and stuff like that. So being able to choose between different metrics, for example, might yield you better results, and Minkowski is just basically any of these matrices at a certain power. So another thing that we were interested in is how well does this correlate with known data. So the idea behind this is that proteins that follow similar patterns are close in space, meaning that they might interact. So one of the things that we thought was, we can just show data that we have of protein-protein interaction and sort of like relate this to distances in Euclidean distances. So on one side of this matrix, we show the distance, which is this blue thing, if I'm not mistaken. Yes, the blue thing is the Euclidean distance, so the darker it is, the closer things are in space. And on the other side, we show annotated data from a data set of protein-protein interactions, where the stronger the brown color is, the more likely it is that the two proteins interact. So there should be some kind of a correlation between the two colors, and in fact, at least here this is darker and it's darker here as well. Here this is lighter and it's lighter there as well. So there is definitely something in the data which correlates together. If we just looked at a single value, if we took the Spearman's R or Pearson's rho, this might actually not yield us any result. It might not even tell us that they are correlated. Why? Because for many of these, of the experimental annotations, there is just no experimental annotation for two proteins interacting. Maybe because they have never been measured. So you are dealing with problems of missing values. So yeah, this is basically three different ways of representing the same data. Let me just go back here. And I think this is the wrong presentation. Yeah. Okay, so another one was, this was my first data viz project with ROSLab, so it's quite a fun one. And I'm actually rewriting parts of it right now, which is also why Burkhard asked me to hold this lecture. So it's the idea that, in biology, oftentimes you do have textual data, you have categorical data. Specifically in this case, we have localization data, and it's annoying. So you look at localization data or data for different proteins, and you go through a list or a CSV file which tells you protein X is located in ABCD. And these names, they are not easy to remember. So I think nucleus is okay, secreted is okay, but then when you start saying like peroxisomal membrane and peroxisomal membrane, whatever not, it kind of annoys you to just read it. So I thought, why don't we just visualize this? And on top of that, show protein-protein interaction data. So it sort of like became more complicated with time. And that's exactly what we are doing. Now I'm just going to open it up for an example. So before I show you that, okay, let me just show you the localization example. So for any protein, I can now see, I can now see that this protein is located, sorry, I clicked the wrong button. Go back. I don't know what I did. Sorry. Oh man.
The internet is something to server is wrong.
Yeah, it's terrible. Oh man. No, it's opening the PPI again.
I guess you could use the LAN.
No, I can't. I don't have a LAN port. Come on. It's 2019. It's not 1994. So yeah, anyhow, the idea here is you can visualize protein location. But let me see if I still have the page open somewhere. Yes. So Okay, so if I visualize protein-protein interaction, this was the reason why we started also overlaying protein-protein interaction data on the visualization. Is you get back these hairballs. There's no way of generating reasonable biological insight from this thing at all. So it doesn't, this is a database, this is literally the database I took the data from for protein-protein interactions. And if you query what does this protein interact with, specifically my example is called PEX19, so one of my 5th proteins, it gives you back this wonderful thing which tells you nothing. Absolutely nothing. So it's like not using anything at all. And let's now see if I can do this here. This is terrible. So I might just try to do this. Then it might be better. Yes. Okay, so if I now seriously visualize this here, I can hopefully both see the thing that I was trying to show you earlier, which is where the protein is located. Ooh, it's located in the cytoplasm and the peroxisome. I hate this word. So yeah, you can see it's located there. It's useful. It's easy to understand. You don't need to explain to people where things are. You can just see it. And then the other thing is, what does this interact with? Well, it might not be necessarily much better than this guy, right? But there is some more insight, meaning that I know now that this protein interacts with stuff that is secreted, it interacts with stuff that is in the nucleus, it interacts with things that are located pretty much everywhere. It has multiple locations, so I can move it around. Now it's in the peroxisome. You can see that, I don't know, any random protein that I pick from somewhere here might have multiple interactions, so there might be cascade events in case of pathways, and I can also overlay a general all-to-all interaction network. Does this make the visualization necessarily better? Well, you know, it's up to the people that look at this. To me, there's an improvement over this guy. It gives you more insight into the biology. But again, this is a very personal thing. Some of the reviewers on the paper agreed. Some of the reviewers on the paper did not agree. Yeah, that's pretty much it. What else? Let me see. So this is just show and tell basically today. Is this the presentation? No, it's this. Okay, last but not least is evcouplings.org. This is an effort by many people. Chris Sander had the idea about 40 years ago. Deborah worked on the mathematical framework, made it possible, and then Thomas implemented it. Oops, this is the wrong slide, but it's the right link. So basically, the idea behind this is biology again. Okay, this is a complex story. So who has heard about couplings and MSAs, profiles? So who has been to PP, to protein prediction last semester? Raise of hands. Nice raise of hands, because I cannot see them. Yeah. Okay. So the idea basically behind the biology in this case is that if we have a stretch of proteins, it's very likely that two positions that are strongly correlated form a contact in 3D. So what does it mean that they're strongly correlated? That if you look at this specific amino acid sequence, and then you take a neighbor in another species, and then you take another neighbor in another species. So you take many neighbors in different species. If all of these neighbors sort of like have these two positions concerned, it's very likely that those two positions are extremely important for the 3D structure of the protein. So I can use those two positions to basically say these two in 3D are going to be in contact, and the rest while the rest can wobble around more or less. So they are used as anchors for the 3D structure of the protein. And from that I can sort of like do de novo prediction of 3D structure. I can start from knowing nothing about the protein structure, just the sequence, and then build the sequence up. Prerequisite for this is obviously that we find enough neighbors which have a similar sequence to my original sequence, but are also different enough so that statistically speaking I can build up this sort of like energy landscape of the proteins. So how do you visualize that? Let me just pull up something. So I'm actually might have this here. Yeah. So this is a protein sequence. It is put out on a ring kind of thing. And as you can see, there's some connections which are basically long connections between, for example, this position and that position. So this might tell us that if you start folding this chain, which is just a straight chain when it comes out from the ribosomes, yes, let's just go for that. It starts folding following, for example, this couple of positions with those couple of positions. They're gonna be in contact, and the rest sort of like follows suit. So you have this obviously these positions which are close to one another, which are also kind of strong, and that might be something like an alpha helix. Yeah, so basically the idea behind this is how can they visualize this without actually visualizing the structure. This is one of the ideas, is having like this, in German you say Pfaden, in these lines between different positions in the amino acid sequence, and sort of like give a visual representation of this. It becomes obviously very complex because you might have a lot of these lines, and there might be a lot of amino acids, so residues on your protein. But yeah, you can basically zoom into any of these positions and see how strong is this position correlated with other positions in the sequence. But this is not the only way of looking at this. Just a second. So this is visualizing the alignment. So this thing that I was telling you about the protein and the different positions that it, if it zooms out. Yeah, unfortunately, another problem with visualizations when you have big data is that they are slow. And basically what you would be able to see here is that some parts of the alignment, like this one, are very conserved. Some other parts kind of vary. And then you can also see that this part is very conserved. So very likely these two parts might form a contact. And this is another way of visualizing the same information that we had before. There's even a better, not necessarily better, but just actually, I don't even know if this is gonna work on Chrome. Oh yes. So what it is doing here is basically just using the colors of, the colors represent biophysical properties of the amino acids. So what I'm seeing here is that there are some lines which are very well conserved. So for example, this one over here, and then I can decide if those lines are all green. I don't know, this is a hypothesis. I don't remember the color scheme, but if green means that they are positively charged, even if there's two green lines which are very well conserved, they're never going to be close in space because they're positively charged. They're going to not attract each other. So you want something that is positively charged and negatively charged. And if you can see two lines of something that is positively charged and something that is negatively charged, then likely those are going to form contacts in 3D. And again, this is a way of visualizing your data without going through the multiple sequence alignments, where the multiple sequence alignment is literally just letters. So you just see, the amino acid sequences. Now this is going to explode. You just see the amino acid sequences, and it doesn't give you any. See, you see this thing, which is at least colored, so it gives you some information. But if there's no color, it's just letters. Kind of useless. Yeah. What else? That's pretty much all of the visualization things that I can think of. Let me see if there's something else. No, that's pretty much it. So I think, so for me the most important take-home message of these slides is basically these guys. Because, I mean, trust me, I've told this so many times to so many people, and they say, I got it. And then you come into a lab meeting, and they do exactly the thing that you said you shouldn't do. So just be very careful, be very thoughtful about how you present data, and do it for yourself, don't do it for other people. So for yourself, lose time in using different graphs, lose time in using different metrics, because it might really just tell you that something is fundamentally wrong with the analysis that you're doing. I have had, you know, I wake up in the morning and I think that there's a cool thing to do, and then I work on it for 2 hours and it works. And I'm super happy and I'm super stoked, and I'm starting to write a manuscript on it, and then I realize, shit, actually, let me try this with a different graph. And then I use a different graph, and it's completely bullshit. So, yeah, you know, I can only tell you from experience, just try as much as you can to visualize your data in different ways, and using different metrics to assert assumptions on your data that you might have, because it might give you a lot of insight about it. Yes?
May I know what kind of tooling you use to produce such a sophisticated graph?
Yeah, so this is a very interesting question. I think it is fundamental for people to be able to validate your findings. So for me, it is important that whenever I do something, scientific or not, it needs to be available on the web, first of all, and it needs to be fast. So there's all of these complications that come into traditional software applications, right? So you saw that there are visualizations. Some of them, actually the alignment viewer, this alignment thing, I didn't write. Because just for me, there's no point in writing something that when you start loading it on a fairly good laptop, it just explodes. So there's no point in visualizing that. There's no one-fits-all answer. I use JavaScript a lot for everything that is web related. I try to push all of the jobs of the rendering of the stuff onto the browser, because it becomes the responsibility of the users to visualize stuff and not mine. But yeah, there's no one-fits-all solution. So if you are doing science and you are, for example, analyzing data sets, what I would suggest is to use just Python notebooks. And for example, one of the things that I wanted to discuss, and I didn't have time to put in the slides, is TSNE, PCA, and UMAP. So who of you has heard of TSNE by show of hands? Sorry, PCA, let's start with the easy ones. Principal component analysis. Okay, TSNE, UMAP. Okay, so this is cool. What is your understanding that PCA is used for? Someone tells me what they think PCA is used for.
It's to reduce the dimensions.
Correct. So you are using PCA to reduce the dimensionality of your data set. So you might have a data set which has 10 dimensions, and then you can project this data set onto 2 dimensions, which has 2 advantages. First of all, it has advantages for machine learning applications, because you might reduce the amount of dimensions, making it possible to use less parameters, for example, in your models. And second, you might be able to visualize the data. So if you go from 10 dimensions to 2 dimensions, you can visualize it on a 2-dimensional graph. I wouldn't suggest to do it in 3 dimensions, because 3 dimensions already, I never showed 3-dimensional visualization, and that's on purpose. There's a lot of people, I think it's about 20% of the human population, that cannot visualize 3-dimensional graphs. They just cannot think in 3 dimensions. So if you show someone a 3-dimensional graph, they see it as a flat thing. I am one of those people, so for me 3-dimensional graphs are pointless. Yeah, so basically what you can do is take your data, which is n-dimensional, and then show it into 2 dimensions. And that might give you some insight visually about what you're looking at. And you can do that, for example, in Python using scikit-learn. Another thing that does dimensionality reduction is t-SNE. It's fairly recent. I think it's 5 to 7 years old. It has had tremendous success with biology because of single-cell sequencing. And it is important to understand why these tools grew in success and with what they are correlated with, because it explains to you particular features of the tool. So in particular, t-SNE is a lot used by anyone today. So if you see people showing you data and wanting to do dimensionality reduction, people tend to, by default, go to t-SNE when there are like many other options, including PCA or UMAP available. But t-SNE doesn't really always make sense. In most cases, it doesn't make sense at all because it has a sense of velocity. So the idea of t-SNE is that you have some data which has a starting point, and then it scatters around in many dimensions with a certain speed. So what you're able to see in t-SNE plots usually, in t-SNE plots of single cell data, is basically a starting point, which is the progenitor cell, and then you start seeing the differentiation of the cell into different tissues, so brain tissue, heart tissue, whatever. So you have this sense of like speed in the data, and you have this sense of directionality. But that is a very proprietary thing of single cell data, and it might not apply to every problem. So you have to be aware that that is one of the limitations or advantages of t-SNE. UMAP does sort of like the same thing of t-SNE, but it is more applicable to just general problems. And also t-SNE is deterministic versus UMAP is deterministic. So if you run t-SNE twice on the same data set, it might, or if you just add another point to your data set, the whole of t-SNE collapses and becomes something different. So every data point in t-SNE, every data point in your data set determines how t-SNE behaves. Versus if you take UMAP, it's a deterministic approach, so it applies a certain calculation on your entire data set, and then every data point sort of like becomes a projection of that distribution. So if you start adding a new data point, it won't change the graphical representation by much. It won't completely break down what you had prior. So that's something that is desirable if you have data that you might throw in and throw out, because otherwise you take 2 t-SNE plots and you cannot even compare them because they're completely different. So yeah, this is just also to give you, I'm sorry that I don't have examples about this, but yeah. And again, all of these things, UMAP, t-SNE, PCA, are things that you can do with Python fairly easy. t-SNE and PCA are in scikit-learn. UMAP is a separate package on Python. And then you can just use matplotlib, which is the default plotting library on Python, to just plot into the points that you get out from the dimensionality reduction.